It's been over 60 years since Watson and Crick described the double helix DNA molecule, and in subsequent years following this critical discovery, our understanding of the importance of DNA has led to technological developments for the life science that have been critical. A timeline of scientific technological development seen here, provides an overview of emergent new tools such as recombinant DNA, DNA sequencing, the use of transgenic knockout mice, the development of PCR reactions, genome sequencing, single nucleotide polymorphisms, as well as numerous others that have actually enhanced and intensified the research efforts in the life sciences. This basically culminating in 2001 by publication of the sequence of the human genome, as well as the completion of several other large projects that have markedly altered the research agenda by actually drawing attention to the importance of understanding the human genome and the effects that nutrition may have on the human genome. In addition, this has led to the human microbiome project that was recently initiated. And why is this important? Because looking at human beings as ecosystems that contain many collaborating and competing species is actually important for the future health of humanity. Therefore, studying the colonization of the human gut has important research correlates to health. Humans on this planet are composed of several cell types. The microbial census exceeds the total number of human cells by about tenfold. And if we were to look at the most diverse and concentrated habitat for bacteria on this planet, it's the large bowel of humans, whereby 10 to the 13 to 10 to the 14 cells per milliliter of fluid reside in that area, comprising thousands of species that make up the gastrointestinal tract microbiota community. The gut microbiota community, in that case, is a complex interrelationship of metabolites and chemical reactions. At least 1,800 genera and 16,000 phylotypes of the species that live in the gastrointestinal tract have been so far identified. And consequent to this, the human host gene pool human to bacteria ratio is something like 30,000 human genes to about 8 or 9 million bacterial genes, thereby showing us that there is a very complex interrelationship of activity. We also recognize that there are 30 or more gut hormone genes that are known to be expressed in conjunction with more than 100 bioactive peptides that are distributed throughout the gastrointestinal tract. Specific non-reactivity of the immune system to a particular antigen, whether this be bacterially or environmentally or nutritionally derived, is an important clue as to the metabolomics of the gastrointestinal tract, because under other conditions, these kinds of molecules can actually produce antigenic activity. And the lack of antigenic activity is the tolerance that the gastrointestinal tract microbiome provides to the human gastrointestinal tract. And as such, we are then in a state of homeostasis.